class, I have what I think will be a really cool lab today. It brings together photosynthesis and chlorophyll that you should have learned about pretty thoroughly in biology, and ties into our organic chemistry unit really well, because we are going to extract chlorophyll from spinach leaves and see it fluoresce. So we're going to excite those valence shell electrons and see that fluoresce. And you're going to answer quite a few questions in the pre-lab to get some background knowledge and also test your knowledge of the procedure, because I know you all read it, right? I'm also going to follow it pretty thoroughly. So I've got all my supplies here. I'll explain them as I go through. And so first up, the procedure tells me to measure about 40 milliliters of alcohol. I have isopropanol here. It could be ethanol too. That'll work just fine. I say about because this is one lab that the measurements don't really matter that much for. So you don't have to be exactly at eye level. About 40 milliliters is perfect, okay? So I'm done with my alcohol, so I can return it to the front of the classroom. I already tore up my spinach leaves in the interest of time, but I had about 6 spinach leaves, and I just broke them into small little pieces. You can tear off the stems, it might make it easier. So the finer the pieces, the easier it is to grind. Notice that I put it in a mortar. Okay, we haven't used those yet. This is a pestle, and this pestle is what we're going to use to grind it up. All right, to make it easier to grind, I'm adding just about like 10 to 15 milliliters of the alcohol. That again is not a very precise amount, but I'm going to grind it as well as I can. The better I grind this, the better my results will be, for the reason that I will break down certain important things in the plant structure to get that chlorophyll off. I've also got some paper towels because this can get messy and chlorophyll does stain. So I want to be aware of that. All right, wipe off those leaves as best I can. Put that there, and then I transfer it to that 250 milliliter beaker. Kind of looks like cooked spinach now. Okay, I don't need to get every little piece, but I definitely want all of the liquid. Okay, notice I'm putting everything on the paper towel as I go. I'm gonna add the rest of the alcohol, and I'm gonna mash it up a little bit more. Okay. And again, I'm trying to get that solution as green as possible. So I'm using a metal spatula, which you're very familiar with, to just squeeze out every bit of that chlorophyll, because that's what we need. Again, please do that for at least like 5 minutes or so if you want really good results. I'm just speeding it along in the interest of time. All right, I've got my filter paper. This is really important because if I just try to use this, I've got a lot of cell parts in there that are gonna, the extra leaf that are gonna block the black light. So I've got a sizable piece of filter paper. To use it in a funnel, you're going to fold it in half like that, make a semicircle, and then fold it in half again. Okay, so now you have like a little cone, like an ice cream cone. Okay, and then I'm gonna open it up and put that inside the funnel. And that funnel, that plastic funnel, is on top of the Erlenmeyer flask, so that's gonna catch the liquid. So then all I need to do is pour this in. It's easier if you mostly decant it. You don't need to shove all those spinach leaves in there, right? That's just gonna require more cleanup later. And you need to let it do its thing and let it filter down. Really only need like 10 mils, if that, in our trusty test tube. So right over here, while that's filtering, I'm gonna make sure I have everything else set up. So I have my flat bottom test tube. These are really nice because if you do happen to set them on the table, they don't fall over as easily. So I like these, and they're a little wider than the other test tubes we tend to use. All right. So I'm gonna try to get as much of that out as I can. Please do not shove your metal spatula in there to try to force it down, because if you do that, you're possibly gonna rip a hole in the funnel, okay? The size of the funnel doesn't matter too much. This one fits the filter paper perfectly, but you might have a slightly different size, and that's okay. All right, I've got most of that liquid down there. Yeah, there's a little bit more, and later if I wanna just decant it off, I can. Or for now, I'm gonna just set that in the beaker so it's aside. And I'm gonna transfer this over to the flat bottom test tube, okay? Have your paper towels ready in case you spill. Notice I got it about 1/3 of the way through. If I had more time, I'd probably try to get it at least halfway. All right. Before this, there were a couple points in the procedure that it said to make your observations. It was important to make observations about the alcohol, what color it was, solid, liquid, gas, viscous, non-viscous, right? All those good things. Make notes about your spinach leaves. And what did those look like? Solid, liquid, gas. Were they thick? Were they fine? You can make some observations about how easy it was to grind them. You can make observations about the yucky paste-like stuff that you were left with. All of those are great. I could have also wet the filter paper a little bit with water like it says. I didn't, just in the interest of time. But that's a good procedure to just make sure the filter paper fits well in there if it's not a snug fit. All right, now I got all those observations down. I'm able to observe the end product. So I have my trusty blacklight here. In class, I would have stopped the video so you don't see these results. These are more for people at home. We're making up the lab, right? So I can see what color that is fluorescing. I will tell you it's much better if you're in the dark. You can see where the light's hitting it, so it's green on the outside and red on the inside. That's because the UV light is only hitting what's in front. Pretty cool. All right, so I'm gonna skip over that part if you're in class with me and just talk about the cleanup procedure real quick. Okay, it asks you, why do you not just dump the spinach parts down the drain? That's really important. In the pre-lab, it asks you that. But all the liquid solution can just be disposed of down the drain. It's totally safe. It's just rubbing alcohol and some spinach leaf extraction. But please make sure you wipe out your mortar, wipe off your pestle, wipe off your metal spatula, rinse them well, wipe out your 250 milliliter beaker, and do not put any solid parts down the drain. So I hope you enjoy this lab as much as I have. I think it'll be really fun for us.